I'm Carlos Ballaré, and I'm a professor of plant physiology at the University of Buenos Aires, and also the National University of San Martin, and also a member of the National Research Council of Argentina. I'm basically study how plants respond to light using specific photoreceptors. My interest in plant science, I always loved plants, basically. That's something natural in the sense that I used to grow plants in my parents' house, a backyard and things like that. So I was always interested in understanding the diversity of plant forms and different colors and different smells. That was something natural. My interest in science came, I would say, later on, because initially I studied agronomy, so I was interested in the production side. I was interested in crop production, but then I somehow got exposed to science. And that I always credit Carl Sagan in the TV series Cosmos as a great motivator. I think he taught me how science works, how the scientific method works, and then I became interested in the scientific method and scientific approaches to questions. But that's something that developed during my time in college. It was not that natural. So in terms of the big questions in my field, I study plant responses to light and how plants use specific photoreceptors to read the light environment. And so I think it's been a long tradition of studying how photoreceptors modulate growth and development. And we are beginning to discover that photoreceptors modulate a number of things that I think we never thought could be modulated by light. And that includes interactions between plants and other organisms, including beneficial organisms, pathogenic organisms, insects and things like that. And this is because these photoreceptors, in addition to modulating hormonal pathways that are related to growth, also modulate hormonal pathways that are related to defense. And I think one of the big questions is try to map the molecular connections between the photoreceptors and these responses that are important for plant adaptation and for interaction with other organisms. I mean, I think a role model is somebody that actually helps you to do better things and to be a better scientist, better in your profession. And in my own field, in photobiology, I always think of Winslow Briggs from Carnegie as a big inspiration because of his enthusiasm for photobiology and for science in general. Also Joanne Corey for her interests in understanding mechanisms. And so there are a number of people that have influenced me through my career. In terms of advice for young researchers, I think the key thing is to be passionate. I mean, if you like a topic, just focus on it and work hard, do your best. And that passion, I think, is essential for science. If you want to have like a 9-to-5 job, science is probably not for you. In the sense that science requires passion and concentration. You also need to be open. As a scientist, you're always asking new questions, you're plowing new ground. So you need to be prepared for setbacks and be open to new ideas. I think that is very important for a scientist to exactly that, to be open, to be prepared for alternative hypotheses. But passion, I think, is the key. In terms of future plans for research, we're interested in understanding how light modulates defense responses, and we're currently focused on the connections between phytochrome, which is an important photoreceptor, and jasmonate signal, which is an important hormone involved in plant defense. And we have discovered a number of things in the sense that phytochrome modulates a number of jasmonate responses. And one of the things that we found recently is that phytochrome modulates, among other things, the volatile profiles, the volatile blends that the plants emit when they are attacked by herbivores. And so it's an interesting thing because light signaling interacts with chemical signaling. So, and that may potentially have a number of consequences for interactions between plants and other organisms in the canopy. And this is something that we would like to understand in more detail, how light signaling, how photoreceptors modulate volatile emissions and the interactions between plants and herbivores and other things in the canopy. And of course, we also try to understand, to get a better understanding of the molecular connections between the photoreceptor, in this case phytochrome, and the important elements of jasmonate signaling. My favorite plant, these days most of our work is focused on Arabidopsis because it's such a wonderful tool. I mean, there is no point in arguing about using Arabidopsis, but I always say that I have a soft spot for a particular plant that I used when I was doing my first experiments as a grad student, which is the Datura ferox. The Datura ferox is called chamico in South America, as a weed of summer crops. It's a very clever plant because it has a number of tricks to synchronize germination or to germinate at the right time, and also use light signals to outcompete their neighbors. And so it's an annual plant. It's a very interesting plant in terms of the responses to the environment. And because I think because it was the first plant I worked with as a scientist, I always, I mean, there is nothing particularly about that, but I have a good connection with this organism.